SFBBO, promoting sustainability by engaging communities for more than 40 years. Our Birdie Hour speaker series is a free virtual program. On May 6, 2026, PhD candidate Trina gave a talk, Wild World of Mating Frog Systems, for our community.
Today's speaker. So this is Trina on the field. And if those of you who joined us back in February of 2024 may recall, she did a Birdie Hour talk on her undergraduate dissertation work, which was using neuroscience to study hearing in songbirds. And here she is with her black-capped chickadee because she was out on the East Coast, not the chestnut ones that we get here. She is currently a third-year PhD candidate in Dr. Jesse Tanner's lab in the Ecology and Evolutionary Biology Department at the University of Tennessee in Knoxville. Her undergraduate degree was in neuroscience and behavior at Vassar, and she is going to be sharing her PhD work on frogs. So I'm going to stop sharing, and I'm going to let Trina take over.
Sounds good. Okay, can you see my screen? Perfect. All right. Well, so welcome again, guys, for joining me. I know it's, a Wednesday evening, and at least for me, being at UT, we're wrapping up our finals season, so things are a little hectic, but I appreciate you taking the time to come listen to me talk a little bit about what I do and frogs and why frogs, especially since this is a birdie hour talk. So as my mom said, my name is Trina. I use she, her pronouns, and I'm currently a third-year PhD candidate at the University of Tennessee. And so, why am I talking to you? Well, first of all, I am from the Bay Area, given that my mom is still there. And my interest in animals was kind of fueled by my family's love of cats. And so we've always had cats growing up, and I've just generally really been interested in animals. For my bachelor's degree, I shipped off all the way across the East Coast and found myself at home at Vassar College, which is a small liberal arts school. I was a neuroscience and behavior major there. And while there, I was exposed to the world of animal behavior research and actually being able to do research that studies why do animals do the things they do. And I was really quickly hooked. And so while at Vassar, I worked in the Gall lab with Dr. Megan Gall, studying sensory ecology in songbirds for about 3 years. And then finally, upon graduating Vassar, I once again shipped off around the country, and this time off to Knoxville, Tennessee, where I'm currently zooming in from, to join the University of Tennessee's Ecology and Evolutionary Biology Department, also referred to as EEB. And as a third-year PhD candidate, I work with Jesse, my advisor, along with other folks within the Collaborative for Animal Behavior to study acoustic communication in tree frogs. So my talk today is mostly going to be sharing the work I've done here at UTK in these tree frog mating systems, but I figured I'd start with a little quick update on where I left off from my last Birdie Hour speaker series in 2024. So briefly, my last talk was focused on understanding the neuroscience of hearing, or more specifically, the auditory processing systems of several different songbird species to understand how these species might be impacted by anthropogenic or human noise. The fun part is that since these talks are fully recorded, you can actually find them on the SFBBO YouTube channel, along with, not just my talk, but all the other great Birdie Hour talks. And so briefly, I found that across these three species that I was looking at, the black-capped chickadees, like the picture I was holding, the white-breasted nuthatches that you see here, and tufted titmice, these three species had different auditory capabilities and noise. That is, the physiology of these nuthatches, of their auditory system, was actually much more vulnerable to noise than the other two species, which might lead them to be more vulnerable to the effects of traffic noise than the other two species. And so just wanted to follow up that back when I was presenting this was all data and I was trying to submit it. I'm happy to share that my advisor and I were actually able to publish our findings in the Journal of Experimental Biology last year. So if you're curious to know more about the specifics of our work, you can look up the article, and I'll warn you that it's a little bit more technical than the talk I gave at my Birdie Hour talk. But if you're ever interested in understanding more about kind of how scientific research works, you can always go look at my article. Okay, so enough about that. Let's get into the field of acoustic communication, how I, as a researcher, study how animals, in essence, talk to one another. And I'll start with birds, because we are obviously all bird people. So, first of all, we typically think of a signaler or an individual who is producing a signal that is then getting picked up by a receiver. So, these are kind of the common terminology that we use. In the field of animal communication, there's a whole bunch of different reasons as to why animals may want to communicate with one another. So, for example, with the songbirds, there's reasons for anti-predator defense, so ganging up on a predator to get them away. You can think of it in flock formation when you're trying to form these groups that are going to flock together. And you can even think of parental care, as my mom kind of mentioned, as, baby birds begging is an example of acoustic communication. And so I'm going to focus a little bit first on this mating aspect of things, right? So when you think specifically of mating, you can think of it as the males typically, although there's some cases of females, but the males who are producing these really beautiful sounding songs that are kind of advertising himself to females and saying, hey, you should come mate with me. And so this is how it's kind of typically thought of. Maybe there's one signaler who's producing a song, that female is listening. But more often than not, there are a lot of males competing for one female. So it's not just one male, but a whole bunch of them that females are often choosing between. And so one really extreme kind of case of this are these sage grouses, which you can see on the right, but they do this really cool behavior, which I'll just play this quick video of. And so what you're seeing are these are all the males who are actually gathering together and displaying. And they do this really kind of funky display where they're like thrusting their chest forward and that makes the things bounce together and it's really funny. And so then all of these smaller individuals are actually the females who are listening to that. So these sage grouses are an example of what's known as a lek, or a type of mating system in which males are displaying and competing amongst each other to try and attract females. The pros of doing this are that A, you're in a big group. So as a collective, it's much easier to attract a female's attention. And also being a group might provide you protection from predation. Of course, the con with it is that you are then competing against other males, but a lot of studies have found that in these lekking systems, the benefits of being in a group far outweigh the costs of being alone. Another unique thing about Lex is that males are only contributing their genetics. Unlike systems in which there's biparental care, so like both parents are sitting on a nest together, there's actually no parental care involved from the male side of things. So oftentimes in these Lex, the males are advertising themselves almost as a sign of their genetics. They are not advertising how good they are as a father, how much territory they have, none of that. In these Lex, it's purely just genetic quality. So thinking back into these bird systems, when you have these lekking mating systems, it's really important for the females to be able to understand the signals, how that relates to the signaler, and to choose carefully because whoever she chooses is going to determine the fitness of her offspring. And so I've, of course, been talking about birds, but I invite you to now think about the same lekking mating system, but with frogs, because this is a really common system in a lot of different frog species. So we have the same thing, a signaler or a male who is advertising to the female with a signal, and oftentimes he's competing against a bunch of other males who are often clustered together in what's known as a chorus. So I'll start off with frogs. What do frogs do? What do frogs sound like? This is a Pacific tree frog, which is kind of native to the West Coast. So you might be familiar with the sound that they make. So frogs are cool because just like birds, you can actually identify which frog is calling based on just listening to them, as each frog kind of is distinct in the way that it calls. Frogs importantly differ from birds in that they're ectotherms, so they're really dependent on their external environment. Just like how snakes and lizards have to bask in the sun, frogs are also dependent on that external environment. And so you can see that the way that they call is by pushing air back and forth between that giant vocal sac and the rest of their body. And so a couple of graphs that I'll have us get kind of familiar with are, as we listen to this beautiful frog again, are first I'll orient you to that the x-axis is time And then the y-axis on the top graph is amplitude, in which case this top graph is known as a waveform. So what you're seeing is that ribbit call basically visualized. And so again, time on the x-axis, how loud that call is on the y. And then the other kind of graph we have is what's known as a spectrogram, where you have time on the x-axis and frequency, or what's known as pitch, on the y. And so the parts that are darker is where most of the energy is in the call. So from these two things, we can look at the calls visually and understand what they temporally look like and what they spectrally look like. And so, as I mentioned, frogs each, just like birds, have their unique calls that allows you to identify them based on those calls. So this is just another example of, again, the spectrograms, which are now on top in the colored parts, and the waveforms, which are the black, of 4 different species of frogs, which includes gray tree frogs, American toads, chorus frogs, and northern leopard frogs. And as you can see, they're very distinct, right? You can tell if you were to just visually look at each one of these, you'd probably be able to see which frog is which. Additionally, you can see that some frogs have pulses, some have calls that go on for really long periods of time. And so in these ways, they kind of differ. And now the cool thing is that these are just one call for each species. But more often than not, males are all gathering in these really big aggregations and forming really, really noisy choruses. So this is an example of a multi-species chorus where now once all the frogs are all together, it can be really hard to pick out individuals. So this kind of brings me to why I study frogs in the first place. Well, they have this really interesting mating system in which these males are gathering together. Females, despite how noisy it is, are still able to kind of listen to all these males, figure out who may be better than the rest, and make decisions about who she mates with. And she does this really quickly. It's been found that females will only listen for about 2 to 3 minutes before they choose who to mate with for the rest of their life. Or, well, not for their life, but at least for the season, who's going to pass on genetics for that round of offspring. So it's a pretty big decision. And so I'm just going to be sharing with you a couple of the projects that I've been working on as part of the Tanner Lab. And so we're going to start off with focusing on the male and the signaler by sharing a little story about variation in signaling behavior. Where might some of that variation come from? Specifically with one of my projects involving spring peepers or Pseudocris crucifer, and how their advertisement calls might signal infection status. So we'll start with the spring peepers. They're mostly in the eastern US. They breed from about March to April. So if you've been around the East during this time, you're probably familiar with this sound that I'm about to play. So their name is probably fitting given the little peeps that they're making. And then again, going over we have got the waveform on the left, the spectrogram on the right. You can see this is a very just one pulse of sound, of which the frequency content is also mostly in this one little frequency sweep. And so as researchers, we can analyze a couple things about this call. We can look at the duration or how long each one of these calls are. We can look at the call rate, so how fast this male is giving this call at. And then we can look at this dominant frequency, or what's kind of the main frequency at which they're calling at. And so why would we measure these things? Why would we look at these things? Well, because females are listening for these things and paying attention as to what they might indicate about the male. So how do these things relate to a male? Well, as I've mentioned, frogs are ectotherms. And so actually in warmer temperatures, they will call faster because they're warmer, they're producing their sounds with their body, and they can do more basically. And so not only is a faster call rate tied with a warmer environment, but it also might just signal having greater energy in general. So I challenge you, if you know any frogs in your backyard, and you know a population that you're passing by quite frequently, you can pay attention to how fast they're calling during a warmer night versus a colder night and see if you notice any discrepancies. Another thing that's indicative of something potentially about the male is dominant frequency is actually linked to age and size. So bigger animals tend to produce lower dominant frequency sounds. And with frogs who have indeterminate growth, so basically means the bigger they get, they don't have a cap on how big they grow. The older frogs tend to be the bigger frogs and thus kind of have the lower dominant frequency. So a female who's paying attention to the spectral content might be able to hear that difference and might be able to indicate, oh, that's an older guy. He may be more fit than someone who's just, formerly from a tadpole, et cetera, et cetera. And additionally, there have been studies that actually found that condition, so body condition, so kind of thinking of like how chunky you are relative to how big you are or how long you are. For males who are kept fed and in good condition, they actually have longer call durations than males who are kept in kind of a poorer diet or unfed. So again, these are all aspects that females listen to because they might signal something about the male and his quality. But one big question in the field is how does infection affect all this? So globally, there's been a lot of diseases that have been affecting amphibians, leading to amphibian mortality. But there's a potential that infection may be actually having what we know as sublethal effects on this aspect of male fitness. So maybe infection is affecting the way a male calls and thus his potential for attracting a female. So there's a couple hypotheses as to the relationship between infection and this measure of vocal performance. The most obvious one that I think most people jump to is that the more infected you are, the worse you feel, the worse you're going to do. So basically, an increase in infection load means a decrease in that performance or how well you're able to do your calling. There's actually an alternative hypothesis in which the more infected you are, the better you perform. And one of the many explanations for this is what's known as the terminal investment hypothesis, where basically, in some ways, because your body knows you're about to die or you're about to, pass away because of an infection, you're like, I need to get offspring as fast as I can and as soon as I can. So there might actually be benefits to increasing your vocal performance to kind of increase your fitness in the short term since you know you're going to die off in the long term. And finally, there's a potential that there's no relationship between these two things, that maybe the method in an amphibian body for dealing with infection is completely separate from vocal performance. And of course, the fun thing about research is that people have found support for all three of these hypotheses in a lot of different species across a lot of different years. And so the field is kind of mixed as to which one it could be. So, in my first year, I decided to kind of test these hypotheses in a population of spring peepers located close to the UTK campus who are naturally infected with chytrid fungus or Batrachochytrium dendropatidis, also known as BD, which leads to chytridiomycosis, which is, again, a global amphibian disease that leads to skin thickening and passing away. So, the question is, are these males who are naturally infected with this BD calling differently from males who are not infected? So during a couple months in 2024, I played a game of Spot the Peeper, which, as you can see, can sometimes be a bit of a challenging task, and basically found males who are naturally advertising, doing advertising calls, and recorded them with microphones. And so we got about 62 males, of which we recorded at least 30 calls per male. Once we recorded those males, we then caught them, took a bunch of body measurements, such as their snout vent length, so kind of how long they are, their temperature, since that's important for understanding how fast they're calling at. And then we also swabbed them, importantly, for BD fungal load, because BD especially lives on the skin of the amphibian. So we can just take little swabs of their bellies, as you can see me doing here, and process those swabs. And so once we collected all this from the frogs, we would let them go back to the ponds that we found them in. And so again, we're analyzing basically what are the call properties of these calling males, what are their infection levels, and seeing if there's any way we can link those two things together. So kind of the first question is, can you predict infection status just based on call properties alone? Can you take a recording of a male and figure out is he infected or not? The short answer is no, but basically I did a set of analyses that kind of looked at all these males, looked at the uninfected versus the infected ones, of which we had about 2/3 of our population was infected and 1/3 was uninfected. And looked at the call traits and said, is there any combination of these things that might allow us to distinguish these two groups as separate from one another? And the answer was no. So unfortunately, we can't use these call recordings as a way to just say, hey, this male is infected or not. In general, we also found that infection didn't really affect male calling. So on these graphs, you have infection load on the X. So now you can see there's variation in how many basically fungal zoospores there were on the various males. And then the graph on the left is dominant frequency. The graph in the middle is duration. The one on the right is called rate. And so for the most part, it seemed that infection didn't really affect any of these traits specifically. But when we actually incorporated body condition or that measure of kind of how, chunky you are compared to your length, we found this interesting interaction in which the males who are in better body condition, so the ones who were probably like eating a lot more, et cetera, the more infected they were, the longer their calls were. And then vice versa, for males who were in poor condition, the more infected they were, the shorter their calls were getting. So it was almost like these good males are getting better and better as they're getting infected, and the worse males are getting worse and worse. So, kind of in conclusion for this, we found that infection was exacerbating these pre-existing differences between the males in good condition and the males in poor condition. And that, funny enough, we're finding, again, mixed support for the various hypotheses that I proposed at the start. And one thing I'm not going to touch on today, but it was also seen in this call duration trait, which all of these calls, there's lots of things you can measure, but call duration wasn't one of the things that we as researchers expected to see a difference in. So it was really interesting that we did. And so kind of future steps in this is to actually test the female preferences to understand if the females can pay attention to these differences that communicate information about infection status. And you might be asking yourself, well, how the heck do we test female preferences? And I'll get to that in the next story. But I did want to share that I was also able to publish this work. And I'm not just putting these in here to self-plug, although of course it's always, I am a researcher and this is kind of my body of work, but just to show how research can be really, really accessible. That when we are publishing our research, we're doing so with the purpose of sharing our science and wanting people to know about it. And so if you're ever curious about a specific research question or think, wow, this finding is really cool, I highly encourage you to go look at a research article associated with it because many of them are super accessible and we're really trying to share our science with the public. Okay, so I've just kind of shared a story about the male and what the males do and how there might be variation in males. But as I've kind of elaborated, it's a whole system. There's not just a male, there's also a female. And understanding how the female is choosing which male to mate with is going to help us understand how selection acts on these male signals as a whole. So for my second kind of story that I'm going to share, I'm going to talk about impacts of variation in signaling behavior on receivers and how that might affect receiver behavior. And more specifically, we're going to shift the frogs that we're talking about, and I'm going to talk about when inconsistency matters and explore the effects of within individual variation in calling on receiver behavior. So we are now going to look at the green tree frog or Hylas cinerea. These are common to the southeast US and they breed from May to August. So they're kind of a little bit later in the calendar year. And so of course I have another video. This one is one that I took with my phone. So sorry for the poor quality. So these guys are really cool. They're bright green. As you can see, they're doing that same pushing the air back and forth between the vocal sac and the body. They're much bigger than spring peepers as well. They kind of sit in the palm of your hand. So to bring us back to our friends, our waveforms and our spectrograms, as you can see, kind of like the spring peepers, they have these single pulse kind of structures, but this time the frequency and the spectral content is a little bit more complicated. So instead of just having a single tone, you can see we kind of have these like three almost peaks, or like a low frequency peak and a high frequency peak that has a couple of these bars within it. So again, here's what they sound like. So you can really link that audio to what we're looking at visually. And as you can hear, A, in that recording, that male was calling a little bit faster than the male in the video was calling. And B, that he also had kind of a little pause in his call rate. So frogs aren't robots. They can't just continuously produce these calls in a perfectly stereotyped way. And so because of that, that introduces these gaps, this little bit of variation that we call within individual variation, or this variation in calling rate for a singular male. And so as I've talked about with the peepers, females are trying to make decisions about who to mate with based on certain traits of these calls. And call rate, it turns out, is actually really important. So females typically like to choose males who are calling at really fast rates. Okay, great, but the question is, how does this within individual variation in this call rate potentially affect female decision making? If you can imagine that this male is kind of on and off, maybe a female comes to listen to him when he's in a bad period, when he's in a period where he's not calling very fast. And maybe because of that, that female kind of writes him off as a not very good male, even if most of the time he's calling pretty well. And alternatively, maybe this whole like, oh, sometimes he calls, sometimes he doesn't, makes it hard for a female to actually estimate what his call rate is. And so, not very many people have studied this, but the story so far in Cope's gray tree frog, or Hylochrysoceles, is some work that was done by my advisor. And females in this species actually care about whether a male is consistent or not. They pay attention to this, and they actually prefer to mate with the males who are more consistent. So, almost like the ones who are more like a little robot that are able to just tick, tick, tick perfectly on the dime. Additionally, female gray tree frogs, their usual choices for faster call rates are actually weakened when you introduce inconsistency into these calls. So it almost acts like a form of noise in which basically females are unable to kind of properly assess and make the usual decisions that they do when they incorporate this variation. And so I was really interested in seeing if this is true for more than just this species. This has really only been found in these gray tree frogs. And within individual variation is important because it's what happens in nature. So there have actually been decades of work in tree frogs and tree frog behavior, but every time we do these studies, for the most part, it's been with calls that have been perfectly stereotyped and kept perfectly controlled because we're trying to control what we can in the lab. But of course, in the wild, things are a lot more variable. So we're trying to incorporate a little bit of that back into the lab. So to the question, how do we even measure female preference? So let's talk about frog biology first of all. So in these species of tree frogs, what typically happens is the males will gather in big groups. They'll start coursing during the summer. When a female is full of eggs and she's ready to lay them, what's known as being gravid, she will kind of find these males, she'll locate them, and then once she decides who to mate with, she'll get close to that male, and the male will hop on her back like a backpack. So in this picture here, the female is actually the bigger one who's on bottom in the dark green, and the male is the lighter one on top. The two will be in this what's known as amplexus position for kind of the whole night, while she drops her eggs, he drops his sperm, and then the next morning they separate and go on off on their merry ways. And so the great thing is when the females are in what's known as this gravid stage, in which they're full of eggs and ready to lay them, they are phonotactically responsible, which just means, or phonotactically responsive, which means that they are physically hopping towards sounds they like. So we can get the females while they're in this period, bring them into the lab, and actually play them sounds, and they will hop towards the one that they like. And they'll do this in the lab, not just the field. So on the right, you kind of see a diagram. On the left, it's a little bit more of a complicated setup than what was used in this experiment, but these are the actual arenas that my lab and I use to test these female behaviors. So I actually have a video from one of my trials. So what you'll see here is basically this is a top-down view. This is our arena. This is where the female is, and I'll play the video and you'll see her in a sec. And I have two speakers playing, one from this side and one from this side. So the idea is we play her sounds, we let her acclimate, and then we let her do her natural behaviors and measure which speaker she chooses. So that's the frog there in the middle. You can see her hop out. She's crawling around. And then once she makes her choice, she just hops towards it. And so a trial ends when a female crosses fully into what we know as these speaker bins. And so that's one trial. And so females are great because we can run them through multiple trials and test lots of different things with each individual tree frog. So, of course, as I've talked about, there's a lot of variation in the way that males call. So you might be wondering, okay, what are you playing from these speakers? So for reference again, here's the audio of a singular male calling. And so, of course, because males are super variable and there's a lot of variability in the way that these recordings call, we need a way to control for that when we're actually bringing it into the lab. And so we are actually able to generate synthetic stimuli. So using the measured properties of the male, so we collected a bunch of recordings and got things like what's the dominant frequency, how long are these calls, how fast are these calls. We can actually use MATLAB and code to generate synthetic stimuli that the females will respond to as if it's a natural male call. And people have done studies to do like, oh, do the females prefer a natural call versus a, synthetic stimuli? And they're 50-50. So the females are recognizing this call just as they would a natural male call. So we can use this in the lab to manipulate everything that we want. So for my question, in which I'm trying to understand, how does this variation in the way that males call affect the female behavior, I designed a set of stimuli that are of these different male phenotypes, in which basically each male has a prescribed call rate, but then his variation in terms of if he's super consistent, then you kind of get a male phenotype that's like this, where it's super consistent. He's always calling at that call rate. Versus you may get a male who's calling a little bit slower and calling a little bit more inconsistently, as in sometimes he's calling fast and sometimes he's calling slow. So for the sake of kind of, illustration, what I've shown you here is there's different call rates. So in red, you have the fast call rates. Yellow, you have the standard or kind of average call rates. And then blue, you have these slower call rates. And then on the left, these are our little perfect, perfect robot frogs. We use a measure called CVW to measure that variation, with the idea being that if you're zero, the closer you are to zero, the more consistent you are, and you don't really have any sort of variation from the mean. And the more variable you are, so it's kind of like an average level of variation and a super high level of variation, the higher that CVW is. So basically, I can manipulate all these males, create all these sound files, and ask questions about which males they prefer over others in response to these two traits. So during the summer of last year, I went out into the field, captured a bunch of these females while they were gravid, ready to be phonotactically responsible or responsive. I keep saying responsible, and gave them a series of trials in which I asked them two questions. First of all, do females have preferences for consistency? Do they prefer males who are more consistent than others? And so what I would do is I would present them with one average guy and then basically present other options against him that differed in that consistency. But importantly, I kept it all at the same call rate. So I'm really just testing, do they have these preferences for consistency or inconsistency? And then my second question was, do females' usual preferences for these faster call rates change with inconsistency? And so what I did is I presented kind of a series of tests in which I'm looking at, what do they like call rate wise when regularity is zero or consistency? And then what happens when you kind of throw that all in? What's going to happen to the female preference? So starting with this first question, do females have preferences for inconsistency? So again, I'm presenting them that black dot represents a standard, and then I'm giving, several alternatives, lots of repetitions of alternatives. And so what you're going to see in this graph here on the x-axis is that difference in regularity. So the idea being that a dot more on the right axis just means that the alternative is going to be more variable or more inconsistent, and then y being the probability that the female chose that one. So if females have preferences for consistency, we should expect to see a line in which the females are more likely to choose these super consistent calls starting up here, and then it would kind of go down. However, we don't actually find that. So we find a pretty flat line. All these dots represent individual trials. And so it seems that at least female green tree frogs don't have preferences for consistency in itself. For my follow-up question, I first had to ask, do females have preferences for call rate, right? So I'm presenting them with, a standard versus a faster alternate or a slower alternate along this whole range. And again, we have a graph in which we see the difference in call rate this time on the X, probability of choosing the alternative on the Y. And this time we see a positive slope. And so basically what this means is that when the alternative is faster than the other one that it's presented with, the females are much more likely to choose it, and then vice versa. Whenever the alternative is slower than the other one it's presented with, the females are less likely to choose it. So in this case, females are demonstrating a strong preference for faster call rates. They are choosing the males who are calling faster. And so then the question is, does this preference change with inconsistency? If we do kind of these same comparisons, but now add in this within individual variation, what happens to that preference? And the answer is not much. So this preference actually does not appear to be changing. It maybe flattens ever so slightly, but statistically, these two values are the same. And so basically what this is indicating is that this within individual variation doesn't seem to matter at all for these green tree frog females, that they're able to accurately assess and still choose the males who are calling at faster rates despite having levels of variability. And so this is kind of a fun finding because, again, for these green tree frogs, the females seem to neither pay attention nor get distracted by these inconsistencies in call rate. But this directly contrasts to kind of the story that we've known so far in the behavior of the gray tree frogs. And so this could be explained by several different things, such as that they have different kind of receiver mechanisms in terms of their auditory physiology system. Maybe they process sounds differently, or maybe it could be due to pure differences in their call rates. So just for example, I'm going to play you, this is what the call of a gray tree frog sounds like. So you can hear it's very different from a green tree frog. The gray tree frog tends to call much slower. It has a lot more pulses in the structure of their call, as kind of seen in this waveform here. So it could be something related to that. And just for fun, I did want to share what our synthetic gray tree frog calls sound like as well. So again, our frogs can't tell the difference basically between the synthetic call and the natural call and are able to make choices even just with listening to computer work. So anyways, future work on this can potentially look at other species, right? We've now only looked at a whopping 2 species. So looking at how these findings might extend to other species would be great to understand what's going on. Is one of the species doing like a really great job at this one specific task, or is there some more broad principle that's going on that we don't know about? Additionally, we can look at other call traits, right? So here I was looking at call rate, but you can look at variation in things such as call duration, or like in the gray tree frogs, how many pulses are in each of those calls is another trait that we could look at. And finally, combinations of other call traits. As I've said, there's multiple different things you can measure, and these different things can differ in very ways. So maybe we need to look at some combination of call trait and call duration to better understand what's going on in the system. And so to tie things back at the start, I've now given you a little story about the signaler. I've given you a little story about the receiver. But of course, there's a whole environment. There's a whole chorus going on. And you might be thinking, what the heck, Trina? Is there a third story? We don't really have the time for this. And this is where I'm going to kind of leave you on a cliffhanger of the work in terms of incorporating what's happening in the chorus. This form of biological noise, as we know it, is kind of to be continued and something that my lab is currently working on. So actually, that picture that I showed you at the start is a project that I plan on running this summer that involves not just 2 speakers, but potentially up to 8 speakers all at once. And we're kind of exploring the effects of choice overload, or what happens when you're presented with a ton of options. In fact, some might say too many options that leads you to make different decisions than you would normally in smaller choice sets. And so this image on the right is an example from one of my advisor's papers that was recently published, in which we presented females with either 2 speakers, 4 speakers, or 8 speakers to simulate different males, which is what is going to happen in more realistic contexts in the wild, to better understand what are females doing. Are they doing the same things that they do in these smaller choice sets? And the answer is no, but you'll have to wait to kind of hear the more complex results of that. So, in conclusion, to kind of wrap up the talk, frog mating systems represent a super powerful model for empirical research. Frogs are cool, even if they are not birds. They're a really tractable system that allows us to ask some really cool questions just about how animals are doing the things that they're doing. They also help illuminate kind of the power of basic science. You know, I get the question a lot of like, okay, so like, what are you doing? And it's really, we're doing basic science to really understand, you know, how animals are doing the things they're doing and just the power of scientific thinking. And then another reason I love what I'm doing is there's opportunities to get a lot of undergraduate students involved. So besides the picture of me and then the picture in the top left, which is my lab manager, these are all different undergrads who I've had the privilege to bring in and work on my projects with. Frogs are great because they don't take a lot of like strenuous training. You can kind of get people in a pond with some waders and a flashlight, and they're able to catch frogs on day one. So I've been really lucky to be able to work with a bunch of undergrads, mentor them, and really get them involved with science and research for opening up potential for future career paths for them. And so with that, I'd like to give a huge thanks to not only my lab and the Tanner lab, but the Velez lab, which is also at UT, Mark Wilbur and Sarah Schrock, who are a lab PI and a master student respectively, who helped with processing the BD samples from my spring peeper chapter, because qPCR is not my specialty. Members of the Collaborative for Animal Behavior, which include Liz Derryberry and Sebastian Stockmeyer, who are on my PhD committee, my cohort from EEB. I'd also like to thank my funding sources, which are a lot of UTK department scholarships, grants from the university as a whole. I'd like to thank SFBBO for having me back, especially since I'm not doing bird stuff. But I'd also like to shout out iNaturalist. We do a lot of our work in terms of finding ponds based on recordings that people take and pictures that people take. So I highly encourage you, if you aren't already on iNaturalist, to get on there. And, if you hear choruses of frogs, don't just think, oh, that's a cool chorus, but maybe take a little recording for the researcher who may need to go into that pond to look for frogs. And finally, I'd like to thank the frogs themselves. I wouldn't be able to do my work without them. And so with that, I'm happy to take any questions. Just for fun, this is a little picture of a male spring peeper that I caught who is missing an eye, and it was really weird. But we recorded him first and then caught him, and it was as we were taking the body measurements that we noticed that he was missing an eye. So clearly, little guy was doing just fine. So yeah, thank you again for listening, and I'm happy to take any questions.
Okay, Trina, why don't you unshare your screen so we can
Yep,
see you? And
sounds
we
good.
do have several questions here. So let's start with, so Taya wrote, what's your favorite frog to work with? And are there any species you hope to work with in the future?
Yeah, I think my favorite frog to work with so far has probably been the little spring peepers because they're so small. As I've seen in some of my pictures, you're really just like they can sit on the back of your hand. And then in terms of the projects that I'm working on in the future, I'll be doing some more with these gray tree frogs or the Hylochrysoceles, because of the interesting things that are happening with that inconsistency. So yeah.
Great. And then we have a question from HW. How long do you have to record in a given area to get the data that you need?
Yeah, good question. So typically what we try to do is we're trying to understand the acoustic properties of a certain population of frogs, of which you can think of like each pond as a population. So what we'll do is we'll record basically each male that we can, that we hear calling, for a minimum of like usually about 30 calls or 2 minutes or whichever one happens first. And so it's like we record 30 calls so that we can capture that within individual variation. Because you could very easily just go like, oh, I'm going to get one call from each male in this population, and that's enough. But we really try to get 30 calls per male. And usually for each population, we try to get at least 10 males. Because as I said, there's some really good males and there's some really bad males. So we're just trying to sample enough in terms of getting a good idea of what the population average is going to be.
Yeah, and another sampling question is, how do you know you haven't captured the same frog again when you're testing different scenarios? Or maybe this doesn't matter, question mark.
Totally. No, that's a fantastic question. And so when we're testing the males, it's a little bit harder to tell because males are basically showing up to a pond like every single night that they can to advertise themselves. Females, on the other hand, are usually only gravid once or twice a breeding season. So we're usually pretty confident that if we're bringing in a female and testing her, when she lays her eggs, once we do release the frogs back. So we don't euthanize any of our frogs. Any frogs that we bring in get released within like about 48 to 72 hours. But we're pretty confident that we're not recapturing a female. We do collect for about 2 to 3 months, usually during the summer season. So it's kind of like summer is all hands on deck to try to collect as many frogs as you can, do as many behavioral tests, and then the rest of the year is spent kind of analyzing that data. There's some techniques known as like toe clipping, in which you clip their toes to signify that you've tested them. But recently, there's been kind of like oh, do we really want to do that? Because the toes just grow back the next year. So you're really only using it as a marker for yourself, like that season, to say, you're clipping their toes. But yeah, so our lab personally tries to avoid the toe clipping, and just kind of focuses on, if we're capturing females, knowing that we're usually only getting one female during her breeding season.
Yeah, and there's a related question, and the person is saying, I don't know much about frog biology, but I'm curious about whether the gravid females that are captured, are they returned to their homes, and does this whole process in your sampling affect their reproductive process in any way, if you happen to know that or not?
Absolutely. Yeah, so good question. So we catch them when they're in pairs, so they've chosen female male, and then we bring them as pairs back to our lab. And then so then during the course of the behavioral testing, we will separate them and the female will hop around, but whenever she's like basically chilling or not testing, she's paired up with her male again and they're kept in the same container. And so sometimes she will lay her eggs in that container because they basically lay this like kind of mass. If you've ever seen a collection of frog eggs, they just lay a whole bunch of them, and it's almost like chia seeds, like all stuck together. But when she lays those eggs, we return them back to their kind of like host populations. So we are not culling kind of the population in any way. There's also like natural selection that happens in which, not all the tadpoles are going to get, survive anyways. Tadpoles are a great food source for a lot of other animals. But no, for the most part, we try to keep them into kind of their natural situations, and then return them back as a pair as well to the pond that we found them in.
Yeah, so the person who asked that question commented, that's really cool. That's good to know. Great study. Okay, we still have some other questions and we have plenty of time to get to them. So Howard asks, are both the male and female tree frogs equally susceptible to the chytrid?
Yeah, so chytrid is really interesting, because so it's like a fungal zoospore that actually tends to live in like the water surface itself. So really these frogs are mostly getting infected just by like existing and living in the ponds that they do. And then it's kind of spread by, okay, if one species has it on their skin and that frog hops off to another pond, then it might infect that pond as well. And so the cool thing about it is that not all species are susceptible to BD. So some of them are what's known as carrier species, where maybe you'll swab their skin and you find like these zoospores, but the frogs aren't sick. They don't fully develop the like the chytridiomycosis, which is kind of the final disease. So it's really a mystery as to why are some species more resistant than others? Why do some never seem to have it? Like you can swab certain species in our area and the tests never show up positive. Additionally, to not only at a species level, but it's also at a population level. So we have some collaborators in Colorado State that we and Colorado State were collecting gray tree frogs and shipping them off to a third collaborator. As part of that, we had to just like sample our frogs to be like, okay, are you infected or not? We knew that all of our gray tree frogs were not infected, but when they tested that batch sample, a positive test came up. So it turned out that gray tree frogs in Colorado were infected with BD, but gray tree frogs in Tennessee weren't. And so it's a whole field right now to try and figure out what makes some frogs better than others at avoiding BD. So long story short, though, it lives in the water, and so kind of once it's infected a body of water, then it kind of infects all the frogs who live there.
Yeah, so we have this great question and sort of a follow-up comment from Joe. So Joe writes, regarding sharing your work, do you or have you considered sharing links to your work with the iNaturalist community in Knoxville? Because that would be a great way for people to realize how valuable, their observations are. And you've kind of, sprinkled in these little comments. And then
Yeah,
he also mentions you looked up the the species in iNaturalist, and there
BD,
really
yeah.
aren't many observations. So thinks that those would be cool to share. So you want to comment on that?
Yeah, no, absolutely. So I've definitely used iNaturalist to find ponds that I'm working in. And so, for example, with the green tree frogs too, it turns out that in Knoxville, we are technically outside of their existing range. So if you look at maps of where green tree frogs are supposed to exist, there's like a suspicious U that dips like around Knoxville, and Knoxville's kind of in the middle of that. So I heavily relied on citizen science of telling people, like when I went to my doctor's office this morning, I was like, hey, if you hear this frog in a pond, you should let me know. And so anytime people have recordings on Iron Naturalist, then I definitely am reaching out to them and saying, hey, I'm a researcher who does X, Y, and Z. I would love, if you have this recording to share with me more information about this, and this is the kind of work I do. a lot of the ponds that we actually do have that we go to pretty regularly, half of them are like wildlife recreation areas that are managed by the town of Knoxville, but the other half are actually people's like private properties and ponds that they have given us permission to work with. So we have really kind of close ties to these various people and are very gracious that they're able to be able to help us. So with the frog stuff, since we look for active courses, it's a little harder in terms of things that we can share. But I can say that one of my lab mates, Ashlesh Patnaik, who works with crickets, he studies this really cool baffling behavior in which they make two little holes out of a leaf, and then they use that leaf to call louder, which is really cool. And he actually created an entire iNaturalist project and advertised that to local groups, saying, "Hey, have you ever seen this behavior?
That
And if
person
so,
has
please."
contacted me. I'm sorry to interrupt, but that's really
Yeah.
funny that you bring that up. Yeah.
Yes. Yep. So he's from my lab. We are good friends. But yeah, just an example of yeah. So he is basically going to use all this data that people are publishing on iNaturalist through citizen science as part of his dissertation. And so once he does that, I'm sure that he'll be sending the results back to that. But yeah, no, we are greatly appreciative of iNaturalist and anybody who really just has their ears out and is listening for frogs, because that's how we tend to find our populations, is just from word of mouth of someone being like, yeah, I have a pond in my backyard. And we're like, we would love to study that pond. So yeah.
Okay, well, we are coming up on time, and we do have time for one last question, which was submitted by Taya, which is, do your frogs get a treat for participating in your studies?
I fear they do not. We keep them for too short of a time to feed them. If we were going to keep them for more than 2 to 3 days, we would be obligated by our what's known as IACUC, or basically our board that sets really strict kind of restrictions to make sure we're not harming our animals or causing them any harm. If we kept them for more than 3 days, we would have to feed them with various things, which I think might include crickets. But since we are keeping them for less than 3 days and then returning them back to their ponds, they do not get any treats, but they do get to be returned back to their male like we found them. So, there's that.
Thank you, everyone, again for joining us for Birdie Hour. Thank you for Trina for presenting. And we hope to see you again at another future event, both the speaker and all of the participants. So take care and have a good night, everyone.
SFBBO is a nonprofit. With your support, we will continue to inform decision makers, inspire people to act, and involve diverse communities in our work. Please visit our website to learn more and get involved today.